Well, so today we are talking about the causative agents of zoonosis and few words about the common features of all zoonoses, because in spite of their taxonomy, location in the classification scheme, they have many similar features. So let us start with that. By the way, just a moment, we'll start firstly with a picture of lesson 9. We didn't discuss spirochetes because you had lecture, but your protocol should be filled in. So I will comment some pictures in the slide. Well, and you will enter and draw the results in your protocols. It is lesson 9 on spirochetes. Well, and you will draw 3 slides of Borrelia recurrentis in blood smear that you already know. You already saw it. It was first lesson in last semester. Because Borrelia spirochetes, and we have shown you these slides. So you will draw it in blood smear. So that is why you can see erythrocytes and you can see coiled bacteria among erythrocytes, purple color. Treponema pallidum, silver in staining, Morozov staining, also known as Morozov staining. You can see corkscrew bacterium with 8 to 14 coils at a small distance between them, unlike in Borrelia. And Leptospira interrogans in animal tissue. You can see it is also silver in staining, because I hope you know that spirochetes are polystained with aniline dyes, except for Borrelia, when we can employ Romanovsky James stain, azurizine methylene blue. Well, for the rest, we can observe them as motile living bacteria in the slide. I mean, untreated slide in a native preparation. We call it native preparation. Or if they are stained, we prefer silver in staining because they become dark colored, and you can see 3 types of morphologies. You know that all of them are spirochetes and they belong to different classes, different categories: genus Borrelia, genus Treponema, and genus Leptospira. That can be different from each other by the number of coils, distance between coils. And Leptospira also have a secondary coils. We call them hooks on the top of cells that make them resembling S and C letters. So you will draw these pictures. Then picture 3, it is venereal disease research laboratory test, used as a non-treponemal test because cardiolipin antigen is employed. It is not treponemal antigen, but it helps to detect anti-treponemal antibodies, reagents, or they look like anti-treponemal. And you will see slide. We're supposed to perform this procedure during class, but if not, you can draw it. And you can see clumps, agglutinates of cardiolipin antigen with patient serum tested that's supposed to be positive, and 2 negative controls. When patient serum is mixed with isotonic solution instead of antigen, and when cardiolipin is checked for the absence of spontaneous agglutination when it is mixed with isotonic solution. Yes, Sheena, yeah, you can submit, but I can save chat as well. So it's not problem, it's already saved. Well, so I supposed to draw the conclusion. It is non-treponemal test, so it is a screening test, diagnosis just to select possibly infected persons. And by the way, do you follow, or you are printing your names? Just draw the conclusion now, Sheena. Well, the test is positive with lipoid antigen, with cardiolipin antigen. So reagents are detected.
Excuse me, ma'am.
Reagents are detected. I mean, reagents in the patient's serum are detected. Reagents are antibodies, non-specific antibodies of against Treponema pallidum. So we suppose that it may be time to repeat.
It's fine, ma'am.
Sorry, it's wrong.
If you want, we can do it another day.
No, just a moment. I'll try to arrange.
We can just like try with another ID if this is causing problems.
Okay, can you see
No.
the screen picture?
No.
No, teacher.
Okay.
Yes, ma'am.
Yes.
Yeah. Okay. Oh. Sorry. So, speaking about spirochetes, the problem of making diagnosis of syphilis is that is because bacteria are poorly cultured in artificial culture media. Actually, they're not cultured at all. Yes, that's why making production of antigens of bacteria to put serological diagnosis is very expensive. And as the disease is diagnosed worldwide, and there are many latent stages in the disease when we cannot see symptoms, serological examination is very important. And it is widely used. I mean, serological tests for syphilis diagnosis is widely used as a screening diagnosis. So to do that, for healthy persons, it's quite enough to perform this test. I mean, VDRL or something like Wassermann test, the complement fixation test. And if it is positive, as a screening test, and if it is positive, we can employ final tests, which are specific treponemal tests when specific treponemal antigen is utilized, which is more expensive. So we can perform it only for those who gave us positive results in the preliminary diagnosis. So what should you draw in your protocols? The results and conclusion for picture 3. VDRL is positive with the cardiolipin antigen. So we suppose that bacterial antibodies to cardiolipin are detected. And we go to the next step of diagnosis. It is also serological. When indirect hemagglutination test is performed with erythrocytic specific treponemal antigen. And you can see, I hope you can see, agglutination of erythrocytes, hemagglutination, until the with the dilution 1:16. What was done? Patient serum is diluted, twofold diluted. Then it is mixed with erythrocytic treponemal diagnostic. The last well stands for control of antigen. We do not add patient serum tested to this well. We just use erythrocytic treponemal diagnosticum. What is it? It is it contains erythrocytes with treponemal antigens adhered to their surface. So it stands for negative control, just to show us the absence of spontaneous agglutination and hemolysis. And you can see it is a button. That is okay.
Excuse me, teacher.
Yes.
I can't see.
Can't see picture?
Yes, I see black.
I see your Google browser.
My Google browser? But you can't see? You can't see picture?
Yes, I can't see picture.
Ma'am, a picture where it's pink color dot. I mean,
yeah,
the
that's
culture
your Google
there,
browser.
And
that's
you can't
what
see.
I'm seeing.
Yeah, me too.
Okay, I'll try. Okay, sorry. I'm leaving just for a second. I'll try to download it again. Okay? Just give me 5 minutes.
It's okay.
Take your time.
So we call them endemic infections. What else we should say about these diseases? They are polypathogenic. I mean, pathogens can cause many infections in different animals. in the whole range of organisms. And in most cases, there is no human-to-human transmission, as I mentioned above. Well, human as an accidental host, it was also mentioned. Endemic and zoonosis can be shared to people from animals, mostly in cases when they have occupational risk of disease. Some occupations are mentioned here: farmers, hunters, furriers, people processing animals' wool, hide, meat, bone, and so on. The next common feature, general property. As they're typical to animals, they lack tissue tropism to human organism. That is why they can invade any tissue and any organ. And clinical picture depends on the location of the disease. We can divide clinical manifestation into different forms. I mean, there is a cutaneous form, bubonic form. Bubo is a inflamed, enlarged, inflamed, and swollen lymph nodes, because they drain parasites from the local area of infection. And many bacteria may survive phagocytosis and may cause even necrosis of tissue and local inflammation of lymphatics, like we can see in plague and tularemia. Later I will show you the pictures. It can be ocular form acquired by contact route when you touch to eyes with dirty hands or utensils. It may be intestinal form if bacteria are ingested, pulmonary, and mostly dangerous septic form. Pulmonary form can be shared from human to human. It is not that end for pulmonary form, pneumonic form of plague and anthrax. So it may cause even outbreaks in a people, a human population. Unlike anthrax, plague, tularemia mentioned above, Brucella usually invades central nervous system and locomotor system. So they do not kill patients, but they cause them, they make them disabled. And we'll talk about the Brucella also today. Well, these bacteria are known as a highly virulent, and they cause life-threatening disease with very high mortality rate, like we can see in septic form, pneumonic form of plague, and anthrax, when it may reach in some cases 100% if untreated, and even properly treated. It may even die. It may even cause death of a victim within few hours after getting the bacterium inside, before symptoms development, like we can see in plague and severe cases of plague and anthrax. And as they are so highly virulent and can be shed very quickly in a population, in a human population, these bacteria can be used as a biological weapon. Speaking about immunity of to the disease, in all these cases, we can say that they are it is cell-mediated in most cases. Unfortunately, reinfection may occur in some diseases, and also such diseases, and we are talking today about plague, tularemia, brucellosis, and anthrax. In all these diseases, type 4 hypersensitivity develops that makes possible allergic skin allergic test, using of skin allergic test for diagnosis, for screening. Well, microbiological diagnosis. Choosing the specimen depends on the location of the disease, on the symptoms. And in all these cases, specimens can be taken cautiously, and they must be placed to approved containers with a secure lid, properly signed, and transported immediately to the laboratory. And working with these pathogens is extremely hazardous, so it should be done only in specialized laboratories, which are specially equipped for working with these parasites. They're biologically hazardous. As for methods, I can mention all 6 that you know, but for some infections, there are methods of choice, of course. Well, they include rapid tests, because infections are very dangerous and can be shared very quickly. That is why genetic tests elicit immunofluorescence used to detect antigens or genome of the bacteria are elaborated. Bacterioscopical examination can be done for preliminary diagnosis and for such infections that that are caused by bacteria with a specific morphology and tinctorial properties, as we discussed before. Bacteriological examination can be performed in specialized laboratories, in equipped laboratories, and for those parasites that are cultured at all. Biological examination, as there are zoonoses, biological examination is also performed, and many rodents, for example, are sensitive to such parasites. Serological examination can be done for infections with prolonged duration, which do not kill victim before we take serum. I mean, in anthrax and plague, serological examination can be performed for well, starting from the end of first week, or maybe beginning from the second week. We can perform them for brucellosis and tularemia diagnosis, but in plague and anthrax, we use serological examination not for antibodies detection, but for antigen detection in a specimen test, like it was mentioned in the rapid tests. And I told you that there is type 4 hypersensitivity that develops in these infections. So patient becomes sensitized to parasites. That is why skin allergic tests can be employed for making diagnosis, at least a screening test. I want to remind you that we never put final diagnosis of infections by the result by positive test with allergen, by positive skin allergic test. But it can be used as a screening test to find sensitized persons among the whole population. To do that, we can use anthraxine for anthrax diagnosis, brucellite for brucellosis, tularine for tularemia, and pistin for plague. Because Yersinia pestis is a causative agent. Plague is caused by Yersinia pestis. Okay.
Yeah.
Treatment is by antibiotics for severe cases of diseases like anthrax and plague, which are life-threatening and can kill patients within short periods of time. There are immune globulins, specific immune globulins. And for chronic cases of diseases like we can see in brucellosis and tularemia, we can employ killed vaccines for treatment together with antibiotics. It is immune antibiotic therapy. Then for prophylaxis in Russia, for example, we use live vaccines. They are not employed for everyone, just for groups of people, for those who live or travel to endemic areas, and for those who have an occupational risk of disease. Such occupations have been mentioned before. Prevention measures include quarantine, wearing protective clothing and gloves, extermination of diseased and dead animals because there's zoonoses, so that animals must be exterminated. Care of domestic animals and pets, I mean by vaccination, and personal and food hygiene. There are common features, common principles of prevention. Now let us move on to the first pathogen to be discussed. It is a member of a family Enterobacteriaceae that was thoroughly discussed before. It is Yersinia pestis, the causative agent of plague. This picture shows you of the microscopical picture. It looks like oval bipolar stained bacteria. It is not endospore in the center, but they are polystained, they're oval, and they are polystained in the center. This is the morphological feature, tinctorial feature of this bacteria. They're small gram negative. Yeah. Okay. Morphology and tinctorial properties. They're small gram negative ovoid, and they show bipolar staining that make them closed safety pin. They look like closed safety pins.
نه میگم زیاد باز نشد به خاطر تنه چه بادیه
Switch off your microphone, please, my dear.
Guys, your
microphone.
Your microphone, yeah.
This is the microphotograph of this bacterium. You can see that they are almost transparent in this sense. Well, so what else? They're non-endosperming. They possess capsule, very big capsule, which is highly protective to them because it protects them from phagocytosis and becomes responsible for generalized protection finally. And they are non-motile, unlike soil Yersinia.
Excuse me, guys. Please turn off your microphone. Yeah.
Thank you. So speaking about cultural properties, some of them are familiar to you because Yersinia, other species of Yersinia, are already discussed. So we can say that they're facultative anaerobes. It means they're not fastidious, but they're psychrophiles. They prefer low temperatures for culturing, and they grow best at interval plus 22, 28, and can even multiply at plus 4 degrees Celsius. They're not fastidious, so they can be cultured in a simple culture media. And what is not typical to Enterobacteriaceae is the result of growth in liquid medium, because being facultative anaerobe, they grow on the bottom, leaving supernatant fluid transparent. Well, then they're lactose negative. That is why they form pink colonies on endo and McConkie agar, widely used for enteric parasites culturing. And blood containing medium maintains the growth of these bacteria, and it lets them grow fast because typically in other media they grow up to two days, but on blood agar they produce colonies within 24 hours of incubation. So their colonies raise in the center, leaving the flat periphery with regular edges. Sometimes they have entire edges of the colonies, and when they look like that, they resemble fried egg, which is typical to Yersinia pestis. And sometimes they form granular colonies, not smooth surface, but granular surface, and they look like hammered copper as the result. They're active biochemically. Oxidase negative, catalase positive, which is typical to all the members of this family. They are indole negative, urease negative, can split glucose as other members of this family with acid and gas formation. Lactose negative, I mentioned it already. And they are negative in Foges-Proskauer test, PP test, that is positive in other Yersinia. So it can be used for differentiation. Foges-Proskauer test is based on the detection of formation of acetylene reaction of Yersinia, typical to other Yersinia. Antigenic properties are due to all antigen in the cell wall, and they possess a whole range of capsule antigens, like F1, which is protective antigen, and antibodies to F1 antigen are diagnostically valuable for making serological diagnosis in convalescence. And VW antigens are also virulence factors of these bacteria, and they're capsule antigens of these bacteria. Virulence factors. Bacteria are highly virulent, and they have many factors to cause disease. They include adhesion factors that can be found in outer membrane of the cell wall. They're proteins, and they possess type 4 pili. They're invasive parasites that can even pass through undamaged surfaces like skin and mucosal cells. To do that, they use fibrinolizin. Well, and of course, they can pass through minor excoriations on these surfaces. Then the invasive parasites, that is why, together with fibrinolizin, they possess neuraminidase to be attached to the target cell, coagulase that causes clotting of plasma, and it causes clotting of blood in the stomach of arthropod vector, rodent flea. Later I will tell you. Lecithinase, hemolysins, and DNase are also used by bacteria for invasion and cell damage. Then, to survive phagocytosis in lymphatics, they use capsule and capsule antigens and TTSS, which is the third type secretory system, typical to all the members of this family. It is a mechanism that helps to invade the target cell. Then they possess extracellular adenylase cyclase that acts as an exotoxin and superoxide dismutase, proteins of Yersinia pestis, and exotoxin, which is lethal to mouse. That is why it is called mouse toxin. It is detected by inoculation of animals. By the way, it can kill rodents, mice, within 1 day after exposure to exotoxin. Then they release pisticin, which is another name of exotoxin, and endotoxin because they are gram-negative bacteria. As for resistance to unfavorable conditions, I can say that they're not resistant. They can be killed by many disinfectants, antibiotics, by heating, autoclaving, ultraviolet light. So they do not live outside the body for a long time. But they can live for a long time in the refrigerator, in the frozen food, in environments. Epidemiology. Zoonosis, plague is known as a zoonosis that is shed from animal to human. And in the next slide, I will show you the principle of the mechanism of transmission. But here, I want to show you the picture of typical scheme of transmission of a parasite from rodents, whole range of rodents, with the red flea or rodent flea as a vector of transmission. It is shed only in pneumonic form. In case of pneumonic form, it can be shed from human to human, and it is endemic area. It is reported in endemic areas as endemic infection. So the typical mode of transmission is transmission via blood with arthropod vector. It is arthropod-borne disease. To be more specific, it is flea-borne disease. Rodent flea is a arthropod vector that can shed bacteria. It is very interesting mechanism of transmission to patient of parasite to patient by rodent flea. After being ingested the flea by the flea, Yersinia pestis multiplies in its gut, stomach, and it releases coagulase. But you know that it is transmitted via blood, so it feeds on the patient, on the animal first, and it eats blood. So it causes coagulation of blood that leads to formation of clotting blood, and it occludes the preventriculus of rodent flea. It is a part of gut of rodent flea between the stomach and esophagus. So formation of clot causes the blockage, I would say, of flea. I mean, it becomes unable to feed on the bitten animal or patient, and to get a meal, it needs to regurgitate the blood into the bite, into the wound, so it can bring directly the parasites from its stomach, preventriculus, inside the wound or in a bitten patient. Then bacterium can also be shed via contact route during processing of animal products through even undamaged or injured skin and mucosal cells. Respiratory route is also possible due to inhalation of dust particles of animals' hide and wool, or with the respiratory droplets from the patient with pneumonic plague. And it can shed in rare cases via elementary route with contaminated food containing bacteria, Yersinia pestis. Well, uh, uh, nowadays we can say that plague is not observed very often, but it is reported every year in some countries, and it was mentioned in the previous slide that it is a seasonal disease. That is when human cases are reported between March and October mainly. But it is known as infamous Black Death in Middle Ages, and we know about 3 pandemics of plague. And the first one was in 6th century, and the second one was pandemic in Europe that caused it population. So it killed many people, and it becomes dangerous to people when there is a crowding of population and when there is a chance to bacteria to shed with this parasite. Well, clinical picture. There are different clinical manifestations of plague, and they include cutaneous form, local damage, the bitten area. We can see inflammation and primary lesion caused by bacteria. Then bacteria go to local lymph nodes, local lymphatics, and bubonic form develops. You can see inflamed lymph nodes, regional lymph nodes. Ocular form is also reported, maybe intestinal, pulmonary, and septic form. All 3 are very dangerous with a high mortality rate, when patient may die within days after getting the disease, after contracting the disease. As for immunity, it is mentioned on this slide, it is mediated, but the infection may occur, it is not highly protective, and type 4 hypersensitivity develops. Microbiological diagnosis. I want to stress that biologically hazardous, and I mean working with living culture. That's why it is basically performed. I mean, examination is basically done in specialized facilities, except for maybe bacterioscopical examination of fixed slides, like we do it during our classes. Well, specimens that can be must be transported in approved containers include aspirates of buboes, lymph nodes, pus, exudate, blood, sputum, cerebrospinal fluid, urine, depending on the clinical picture, autopsy material in fatal cases, and animal specimen, because bacterium is shed from animals, and animal samples can be tested or must be examined as well, including arthropods as well. Well, and all 6 methods that you know are mentioned here, because rapid methods can be performed, because we need to put the diagnosis very fast to stop the transmission of parasites, to prevent transmission, to start quarantine measures. Bacterioscopical examination is also possible because bacteria have a specific morphology. Closed safety pin appearance makes possible to suppose the presence of Yersinia pestis in a specimen tested. Bacteriological examination is also possible because bacteria are easily cultured in artificial culture media. In some cases, biological method can be employed because bacteria can be cultured in rodents, and due to the presence of lethal exotoxin, lethal to mice, we can use it for rapid diagnosis, not rapid, to diagnose disease in specialized laboratories, using mice as a model for inoculation, because it will die within 1 day after exposure. Serological examination, look, is used only for antigen detection during course of disease, or for antibodies detection against F1 protective antigen of capsule in convalescence, to put the diagnosis after recovery. And skin allergic test is not widely used, but possibly used. It is based on the decline of pistin. By the way, don't confuse pistin with pisticin. Pisticin is a bactericidal substance produced by bacterium, and it is not allergen of bacteria, but pistin is made of bacterial cells. Well, here's a scheme of bacteriological examination. It looks like that. Specimen tested is inoculated on the culture medium. You can see fried egg appearance in the picture. By the way, optimal temperature is mentioned on the slide. And for rapid diagnosis, we one more plate is chosen, is selected, and it is inoculated with a mixture of specimen tests, like pus of bubo, exudate of bubo, mixed with bacteriophage, specific bacteriophage. And next day examination shows you the appearance of lysis of culture, which shows us the presence of Yersinia pestis in a specimen test. Identification of bacteria is based on the detection of morphology tinctorial properties, agglutination with specific antibodies. It is not shown here, but biochemical testing is also performed. And again, bacteriophage susceptibility test is employed, as you can see it here. Well, treatment and prophylaxis. Treatment includes antibiotics, immune globulin for severe cases that can kill victim within a few days of disease after exposure to a parasite, and immune prophylaxis is by live vaccine, and it is employed for people living in or traveling to endemic areas and people having occupational risk of disease. Prevention includes deratization, extermination of rats and other rodents living in our houses, dusting houses with insecticides to kill arthropod vectors, rodent fleas, and avoiding exposure to rodent flea bites using insect repellents. Of course, for people that work in the laboratory with these parasites, protective clothing, gloves, glasses, masks, not like that, of course, in the picture, shown in the picture, are also very necessary. You can follow any information on coronavirus outbreaks. You can see the garments used for these purposes. Yes.
Ma'am, but what about the beak?
About what?
Beak.
Beak. It is a picture of plague doctor. You can Google it in internet concerning the outbreaks of plague in Europe. And it was thought that using this mask with beak, by the way, that is filled with the plants with bactericidal effect, it was believed that it can protect from infection from outbreak of disease. Nowadays, by the way, in Venice, every year in February in Venice, there is a tradition to use such masks, maybe in memory, to the memory of these outbreaks, outbreaks of plague in Europe that killed many citizens of Europe.
Miss?
Yeah.
Can you get like a secondary post-exposure?
Secondary, you mean what?
A secondary post-exposure, like for example, you already got it once and like you can get it again. Like, is the vaccine like for like some months or some years? Or is it like life?
Duration of immunity after vaccination. It is believed that live vaccine can protect for several years. As for vaccine against plague, it is not highly protective, and it maintains high level of antibodies up to up to half of year, up to 6 months, up to 1 year. Well, so for people having occupational risk, like working in the laboratories, laboratory workers, laboratory staff, they should be revaccinated by booster injection every year. So as for infection, unfortunately, bacteria have protective antigens. Exotoxin is protective, and F1 antigen is protective as well. But the duration of immunity is not very long because antibodies that develop are not protective. That's why, reinfection may occur, as it was mentioned before. But vaccination is effective enough, yeah.
So we can't get, so we don't develop like secondary antibodies to it, like
secondary antibodies. You mean IgG?
Yes.
They develop as a according to the typical process, because primary infection gives a high level of IgM, but reinfection and later they are replaced by IgG, of course.
No,
right?
no,
Yeah.
I meant
Yeah.
like, for example, like since we have a risk of getting reinfected by the disease, like we don't develop like a secondary immune system for it. Like for other disease, like the response is so high that we mostly don't get the disease because our body is responding to it. So for this, we don't develop.
So it is
like
not unfortunately highly protective.
immunity.
So reinfection may occur, maybe not in many cases, not very often, but it is reported, unfortunately.
It is possible.
Maybe because it is cell-mediated immunity, that's why. That's why.
You.
But fortunately, we have treatment, specific treatment. Antibiotics are helpful enough. By the way, it is very typical to severe parasites. They are not sensitive. They are not resistant to environmental conditions in many cases, and they are killed normally with antibiotics. I talk about bacteria, of course, not viruses. Anything else on plague?
No, thank you.
Okay, let us move on to another gram-negative bacterium. By the way, it causes infection with the similar symptoms to plague symptoms. And the disease is named Tularemia. It is caused by Francisella tularensis, named after Francis. Tulare is a geographic region in America, if I remember rightly. So it belongs to family Francisellaceae, genus Francisella, species name Francisella tularensis. Under the microscope, it looks like very small bacteria, like a dots. You will draw it like a dots today. Well, and they are described as a small gram-negative ovoid bacteria, ovoid rods, or even coccobacilli, very short. Non-endospore forming, non-motile, may possess microcapsule. By the way, the presence of capsule and microcapsule is typical to many bacteria isolated from living host. So in freshly isolated cultures, in many cases, we can say capsule, encapsulated bacteria, and they have type 4 pili for attachment, responsible for adhesion. Well, the aerobic.
Excuse me, ma'am.
Yeah.
So there is capsule is added, right?
Yes, they have a capsule. Yeah.
Okay,
it is very
thank
small, the narrow capsule, so we call that microcapsule. But it surrounds cell wall immediately, so we can call it capsule or microcapsule.
Okay.
Doesn't matter.
Okay, ma'am. Thank you.
It's aerobic. It prefers CO2 atmosphere, CO2 presence in atmosphere, so we can mention them as capnophiles or microaerophiles. They prefer low. It means they prefer low concentration of oxygen, but need CO2 in atmosphere, so we can call them capnophiles as well. They're very fastidious. They're not easily cultured in artificial culture media. Grow slowly up to 5 days on highly enriched media containing cysteine, blood, egg yolk, glucose, serum. And for making diagnosis of tularemia, by the way, scientists, laboratory workers start with inoculation of animal model or chicken embryo. And after that, they take specimen from living model and then put it to the culture medium, because they are not primarily isolated on the solid medium, even highly enriched. Well, so we can say as well that they are facultative intracellular parasites. They survive phagocytosis and they prefer living in the host, in the living cells. That is why as a models we can use rabbits, because these bacteria are highly pathogenic to rabbits. And we can use chicken embryo and we can inoculate it into egg yolk sac. Working with these bacteria is extremely hazardous as well, so we should be performed in specialized laboratories. Well, biochemical features. As the intracellular parasites, very fastidious, they have no high activity, biochemical activity. They utilize glucose with only acid formation. They can destroy dextrin. They can form hydrogen sulfide, and they possess phosphatase and glycerol activity. Being gram-negative, they have O antigen in the cell wall, outer membrane of cell wall, and they also have VI antigen, which is similar in structure and composition and activity to Brucella. That will be discussed after Tularemia causative agent. Well, virulence factors, not many. You can see adhesion factors type 4 pili and outer membrane proteins responsible for attachment. They survive phagocytosis, so they have capsule for these purposes and lipopolysaccharide. And as they're gram-negative, they possess endotoxin. Give me this, my papers, sorry. Well, resistance to unfavorable conditions, resistant to low temperatures and moisture, can be killed by heating, autoclaving, pasteurization, and antibiotics, disinfectants. Nothing specific. It is against zoonosis. There is no human-to-human transmission. Human is a dead end. It is endemic. The main route of transmission is transmission via blood with arthropod vector that includes ticks and mosquitoes. It can be shed with respiratory route, via respiratory route with dust particles, aerosols, contact route during processing of animal products, and sometimes it is transmitted, it is shared with via fecal oral route as well. So the main route of transmission is via arthropod vector. But as I mentioned in the beginning of today's lecture, all zoonoses can be shared via all three modes of transmission. Tularemia, you can see symptoms similar to that of plague discussed before. You can see bubonic form with enlarged lymph nodes, regional lymph nodes. But unlike in like plague, tularemia has a prolonged duration with a longer incubation period, prolonged duration, and in most cases it is not fatal. It is not life-threatening. But there are fulminant forms of tularemia when high concentration of microbe causes disease. In this case, it can be fatal. And you can see the prolonged duration makes prolonged immunity. There is a protective prolonged immunity. Type 4 hypersensitivity develops. Antibodies are protective and they can be detected serologically to put the correct diagnosis. Here's ocular form of tularemia. Specimens like in plague include whole range of specimens depending on the clinical picture. In fatal cases, autopsy material can be tested, animal specimen because it is shed from animals. But as serological examination is the main method of diagnosis, don't miss blood serum as a specimen tested. Well, for among methods, of course, I can mention rapid methods, immunofluorescence, PCR, bacteriological examination that is performed after biological method, I mean, after inoculation of living model. And then isolation of culture is possible in artificial culture media. Serological examination is the main method, and skin allergic test with tularemia is used as a screening test. As for serological examination, tests used for serological examination, we can mention here agglutination test, indirect agglutination test with diagnostic titers of 1 to 100, indirect immunofluorescence test, which is a very sensitive test used for making diagnosis. Here's a scheme of diagnosis. Patient serum is taken. It is diluted, twofold diluted, and diagnostic titer is mentioned in the scheme. And today we'll put down the result of of the test in the protocol a bit later. Biological examination is shown here. A specimen tested is inoculated into animals after making animals diseased. We're taking specimen, inoculate culture media, isolate culture of bacteria, examine morphological and territorial properties, perform slight agglutination test. It is done for making diagnosis of tularemia using biological models, and serological method is found here as well. And open your protocols, find table 20, and let us enter the results of test of serial dilution agglutination test. The procedure is the following. Do you have it?
Yes, ma'am.
Yes. Okay, Paramita. So here's a scheme of this test, is the procedure. Patient serum was diluted. Twofold diluted started from, it was primarily diluted 1 to 25, and then twofold dilutions were prepared, 1 to 50, 100, 200, 4, 1800 respectively. There are 2 tubes that stand for control. After preparing dilutions, by the way, the diagnosticum, tularemia diagnosticum is added. 3 drops of diagnosticum are added to the test tubes and to antigen control tube. So it is agglutination test. It means particulate antigen is utilized. And when we bring it to isotonic solution for control, we can see turbidity. We can see turbidity here because particulate antigen is added and it's negative result. In antigen control, there is no antibody. There is an antigen mixed with isotonic solution. Serum control contains patient serum tested in the primary dilution 1 to 25, mixed with isotonic solution. Patient serum is added without isotonic solution, so it is dilution 1 to 25. And you can see transparency. It should be maybe a bit pale, a bit yellowish because it is a serum. Oh, it's snow outside. Okay. Well, and then after 24 hours of incubation, we can see the result. Antigen control is negative. You can see turbidity, cloudy. It is cloudy. Serum control transparent. And you can see first 3 tubes with flakes due to positive result. It means there is agglutination in the dilution 1 to 50, 1 to 100, 1 to 200. It is labeled with pluses, positive result. In the rest 2 test tubes, we can see turbidity like an antigen control. It means it is negative. The conclusion, agglutination test is positive. The titer of antibodies against antigens of Francisella tularensis is 1 to 200. And I'll remind you that diagnostic titers start from 1 to 100. So it is twofold higher. And you can say that diagnosis of tularemia is confirmed. Do you follow?
Yes.
Yes, ma'am.
Okay, Sheena. Should I repeat something? No? If everything is okay, we'll move to the next question. Should I repeat? It's a commonly used reaction. You know, we already discussed serial dilution method. You discussed it in the previous semester, right? It is standard agglutination test or tube agglutination test. It is serial dilution method when patient serum is diluted and mixed with the diagnosticum. So patient serum contains unknown antibodies. Diagnosticum contains known antigens. Known antigen today is tularemia causative agent, Francisella tularensis. And it is positive in first 3 tubes because you can see flakes here. It is a positive result. It means diagnosticum antigen binds to antibody and it forms flakes finally. If negative, you can see turbidity due to the presence of particulate antigen. That's a common rule. Treatment includes antibiotics in prolonged duration of disease, subacute disease, many chronic case of disease. Killed vaccine can be employed together with antibiotics. The principle is similar to that of treatment of chronic cases of gonorrhea. Bacterium is found intracellularly inside the target cell, and antibiotics cannot reach bacteria inside the target. That is why we inject vaccine. Killed vaccine, it is not live. It contains killed bacteria, and it lets bacteria outside the cell. Once being outside the host cell, it is killed by antibiotics. That's a good practice known as immune antibiotic therapy. For prophylaxis, we have live vaccine, and it is given to people living in endemic areas, so maybe traveling to endemic areas, like it was mentioned in plague, and for people with occupational risk of disease. Well, prevention, I told you that tularemia is similar to plague in many items, many characteristics. So prevention also includes deratization, avoiding exposure to insect bites. That includes insect repellents and protected clothing in endemic areas, in forests, for example, where you can be accidentally bitten by a mosquito or tick, because it is tick-borne and mosquito-borne disease. Questions on tularemia? Did you draw the result of agglutination test?
Yes.
Yes. Let us move on to brucellosis. It is also caused by gram-negative bacterium, Brucella, that belong to genus Brucellaceae, named after Bruce. Genus Brucella includes many species, and the typical human parasites are actually not typical human parasites, they're animal parasites, but typically diagnosed in human cases are three species: Brucella abortus, Brucella militensis, and Brucella suis. Brucella abortus is shed from cows, Brucella militensis from goats and sheep, and Brucella suis from pigs, which is swine Brucella. They're small gram-negative rods. They do not have any specificity under the microscope, and we can easily confuse them with Francisella and Brucella under the microscope because they're small gram-negative, avoid rods and Coccobacilli, nothing specific. You can see the scheme. It looks like Francisella mentioned before.
Excuse me, ma'am.
Yeah. Yes.
Ma'am, Swiss is from whom? You
what?
said one is cow, another is sheep and goat, and the
Okay.
last one?
Brucella abortus causes septic abortions in cows. So it is shed from cow,
Yes, ma'am.
human, from cow to human. Militensis is shed with goats, from goats and sheep. And Brucella suis is swine Brucella. It is shed from pigs to human, of course.
Thank you, ma'am.
You're welcome. So they're pleomorphic. It means they may form cocci or bacilli. They're non-spore forming, may possess microcapsule, non-motile, like for Francisella discussed before. Aerobic microaerophiles, some of them require, Brucella abortus requires CO2 in atmosphere, and it belongs to capnophiles. Differentiation of species of Brucella is based partially on the ability of bacteria, detection of the requirement of bacteria to CO2 in atmosphere. Grow slowly, look, up to 3 weeks. And blood culture of a patient with suspected brucellosis must be kept in the incubator up to 1 month prior to detect the presence of bacteria. So they grow slowly, slow growing bacteria, very fastidious, require specialized enriched media, form very small smooth colonies with nothing specific in their appearance. They are described as a pod-like sometimes. Like Francisella, they are facultative intracellular parasites, so they can be cultured in animals, rodents, and in chicken embryo inoculated into egg yolk sac. Scientists work with these bacteria in specialized laboratories, and microbiologists, not only scientists, but bacteriologists work with these bacteria in specialized laboratories, especially equipped for working with intracellular parasites. It's just a moment. Biochemical activity is not very high. Actually, it is actually low activity because they do not split sugars typically, but some of them may release hydrogen sulfide. It is for Brucella abortus. It releases hydrogen sulfide as a result of proteolysis. And in my biochemical tests, I need to mention oxidase activity, catalase activity, ability to utilize glucose with acid formation. Oops, in Russian, acid. K stands for for кислота in Russian. And they possess urease activity. Look, tests used for differentiation are the following. We can differ between them by the ability to produce hydrogen sulfide for Brucella abortus, by the requirement of CO2, Brucella abortus again, and bacteriostatic effect of aniline dyes. It is not actually a biochemical test, it is a like bactericidal effect, bacteriostatic effect. It's like antibiotic susceptibility test, but instead of antibiotics, aniline dyes are employed, like basic fuchsin and cyanine. And here's a picture with a plate for Brucella, seeded with 3 strains of Brucella. Brucella abortus, Brucella militensis, and Brucella suis. They applied as a sticks. It was sticked with the 3 species of Brucella. Then it was covered with 2 filter paper strips impregnated in fuchsin and cyanine. Both are aniline dyes. And you can see that Brucella abortus can grow in the presence of fuchsin, so it is not inhibited, and cannot grow in the presence of cyanine. You can see no growth. The opposite result is for Brucella suis. It cannot grow in the presence of fuchsin, but can grow in the presence of cyanine. And Brucella militensis is not sensitive to fuchsin and cyanine, can grow in both cases. So it is an easy test used for their differentiation. I'll repeat that it is a plate with culture medium for Brucella, sticked with 3 species, and you can see no growth under the strips for Brucella suis with fuchsin or Brucella abortus for cyanine. And you can see that Brucella militensis is resistant to aniline dyes that you can see on the bottom in the table.
Guys, please switch off your mic.
Thank you. Antigenic structure of Brucella. Being gram-negative, they possess O antigen. And it was identified that O antigen of this bacteria contains 2 subunits, consists of 2 subunits, A and M. A stands for abortus, M for militensis. And combination of these subunits is used, detection of prevalence, I will tell in other words. The detection of prevalence of subunit A and M makes possible to differ between species of Brucella. But that fact that all species of Brucella, any species of Brucella, contains both subunits A and M, helps to develop immunity against all possible sorts of Brucella. I mean, as any Brucella has O antigen similar in composition with the presence of A and M surface antigens inside, makes possible to prevent disease against all different sorts of Brucella, because all of them are similar in structure and they differ only in the prevalence, in the concentration. So, I mean, A and M antigens, components of all antigens, can be found in different ratios in all species of Brucella. That is why vaccine against Brucella is made of single species.
Guys, can you please switch off your microphone? Like no one can hear. Guys.
Okay. May I continue? So vaccine against brucellosis is made of Brucella abortus. Mute your microphones, please.
Sarah, please. Zara Parsi, please. Hello. Guys, are you
زهرا
listening?
پارسایی عزیزم میکروفون تو ببند.
Hey, they're in their own mood.
May I have my words? Sorry. Okay.
So sorry, ma'am.
Virulence factors include byline for attachment, invasive factors, hyaluronidase, that is, by the way, important to bacteria to cause damage of connective tissue, Then, being gram-negative, they possess endotoxin, and they're facultative intracellular parasites and they survive phagocytosis. To do that, they possess microcapsule, and they can inhibit formation of phagolysosome. So it is also antiphagocytic factor because formation of phagolysosome, that phagosome fuses to lysosome with rich in lysosomal enzymes, is either one step of phagocytosis, one of the steps of phagocytosis. Resistance, resistant to low temperatures, but easily killed by heating. That is why proper cooking can kill these bacteria very fast. Well, epidemiology. I already mentioned the sources of infection to human. It can be cow, cattle, it can be sheep, swine, pigs, and goats. So the source of infection is livestock. So it is typical zoonosis, and there is no human-to-human transmission in brucellosis. Human is a dead end. The main route of transmission is a fecal-oral route. It is typical foodborne that is shared by consuming improperly cooked, undercooked meat and unpasteurized raw milk and maybe cheese, soft cheese made of raw milk, unpasteurized milk. Because heating, proper cooking, boiling, pasteurization kill the bacteria very quickly, within 5 minutes. Well, it can be also shed via contact route and respiratory route when you pet animals or when you process some animal products. And that is why infection is mainly observed in people having livestock or working with the parasite, working with animals. Pathogenesis. You can see the picture, the scheme that shows us possible way of inoculation, way of transmission of a parasite. And unlike Tularemia, plague, and anthrax, this infection has some has another steps of pathogenesis. And portals of entry include gastrointestinal tract because it is mainly shed via fecal-oral route. It can shed, it can enter the body through skin and mucosal cells, conjunctiva. Here, portals of entry. Bacteria survive phagocytosis. They multiply in macrophages, and in macrophages it reaches bloodstream, causing bacteremia, and bacteria circulate in blood and remain in internal organs, causing damage of central nervous system and locomotor system. And it can be shed with excreted with milk in animals, and it can be found in breast milk as well. So breast milk can be used as a specimen for making diagnosis of brucellosis. Well, clinical picture involves damage to a locomotor system, and we can observe symptoms of arthritis and arthrosis, and maybe severe damage to cardiovascular, severe damage to heart with cardiovascular complications, and very dangerous neurobrucellosis is very dangerous because it can cause symptoms of meningitis and even encephalitis. But it starts with the non-specific flu-like symptoms. That is why the primary steps of disease can be missed in misdiagnosis, and it may cause severe complications with disability development. Fortunately, it is not fatal in most cases. The mortality rate is lower than 1%, but patients become disabled. Immunity, you can see the new term, non-sterile immunity. Actually, it is not new because the similar sort of immunity was discussed in tuberculosis topic. Because while bacterium is inside the body, patient is immune. Person is immune to reinfection. Well, like we can see in patients immunized against tuberculosis with BCG, or having primary lesion in tuberculosis on the top of lungs. In here, the similar principle. Once patient is infected with Brucella, he or she becomes resistant. Once we eliminate the parasite, person becomes sensitive again. Immunity is mainly cell-mediated. Type 4 hypersensitivity develops, and as it is non-sterile immunity, reinfection may occur when bacterium is eliminated from the body, completely eliminated. Microbiological diagnosis. You can see specimens that include blood, because there is a circulation in blood, there is bacteremia. Well, urine, cerebrospinal fluid, synovial fluid as joints are invaded, bone marrow aspirates, breast milk can be examined, autopsy material in fatal cases, and blood serum for serological examination, because serological examination mentioned here stands among the main methods, the prevalent methods. Well, other methods, rapid tests, bacteriological. You can see bacterioscopical is not mentioned because there is nothing specific in morphology and tinctorial properties. Biological examination by inoculation of rodents or chicken embryo egg yolk, sac. Skin allergic test with brucellin, it is known as a Burnett test, is mentioned here. And in the next slide, we will stop on serological examination, but I want to comment some pictures in this slide. Here's a result of Burnett test. It is skin allergic test when brucellin is injected intradermally, like we in a similar way like we discussed in tuberculin test. The similar principle. Intradermal injection helps to see inflammation and swelling in the area of injection, and the diameter of papilla and redness is measured. And brucellin test is positive in patients with the current disease, in convalescent people, and vaccinated people. So in any case, when person is exposed to the parasite due to vaccination, due to current disease, or due to infection in the past, we can see positive test. So like tuberculin test, it is used for screening only, because we can't put diagnosis, final diagnosis by this result. We can simply select persons possibly infected. Well, this one is agglutination test. I will comment it next in the next slides. And this one is a very interesting test, I think so, that is used for detection. How to say? It is used for detection of milk taken from from diseased animals, from animals with brucellosis. I already mentioned, by the way, it is not shown here in this slide, but you must keep in mind that in zoonosis, animal specimen is examined because it can be used for processing, for preparing foods, for making bone meals, for preparing, well, for sewing, for processing hide, for making leather, well, for making shoes, everything. So when it is made of animal products, it should be tested for the possible contamination with the parasites. That's why milk taken from diseased animals is dangerous, and we need to know whether it can be employed as a raw milk or it should be pasteurized. Can we make it is important to decide whether it is used as a raw milk for making soft cheese, for example, or it can we can drink it without heating, or we need to do something before consuming. So what is the principle? Milk is contains secreted antibodies, IgA. Well, when it is taken from animal with infection, it will contain antibodies against Brucella. So what is the procedure? It is just agglutination test with Brucella diagnosticum. So we have two portions of milk tested. First three is from one animal, the next three from another one. So milk is mixed with Brucella diagnosticum. So it is antigen antibody reaction. When it is taken from animal with infection, the purple ring is found above the layer of milk. It means antibodies IgA of milk against Brucella bind to Brucella diagnosticum, Brucella antigens, and they form antigen antibody complex above the layer of milk. This milk is taken from diseased animal. I mean, it cannot be used as a raw milk. It cannot be consumed without heating, without pasteurization. In the left three tubes, we can see white ring, and you can see that sample becomes uniformly purple because Brucella in diagnosticum are not catched by antibodies, and they are distributed uniformly in the layer of milk. The white ring above is a milk fat. So this milk is safe. It can be consumed without heating. Is that clear? This one is PCR result. Well, open your protocols, table 21, where you can see scheme with the Huddleston test. Huddleston test can be used as a screening test. It is slight agglutination test, but performed as a quantitative analysis, for quantitative analysis. Here's a scheme. Patient serum is diluted with the help of antigen, Brucella antigen. Brucella are very small in size. That is why in laboratories, Brucella are usually colored. Brucella diagnosticum usually have color to make the result visible, easily visible, because they're small in size. Well, patient serum in different amounts was mixed with the same volume of antigen. You can see antigen is added by 0.003 milliliter to each quadrant. And serum was added in different amounts, starting from the biggest one, 0.08, up to 0.01. So this is the minimal dilution, 1 to 50. Then twofold less, 100, 1 to 200s, and 1 to 400s. In the control wells, we have serum control that contains serum in a volume 0.03 mixed with isotonic solution instead of diagnosticum. An antigen control added in the volume 0.03 milliliter with the same volume of isotonic solution instead of serum. What do we have? In a serum control, we can see transparent fluid just for the absence of spontaneous agglutination. In the antigen control, we can see turbidity. We can see particulate antigen of Brucella diagnosticum. It is colored, pink in color. And I will tell you that Huddleston test is estimated as positive when we can see positive result in all 4 cells. I mean, until the titer 1 to 400s. And you can see agglutination here, here, and here, and in here also. You can see clumps in all 4 cells. So we can say that you can label it, by the way, with pluses and minuses. And we can say that slight agglutination test is positive until the titer 1 to 400s. Until the titer 1 to 400s. It looks like that. In here you can see positive in only 2 cells. The rest are negative. But we have 4 positive results. What does it mean? It is estimated as positive. This person is suspected as a patient, and we need to perform the next step reaction. It is Wright's test. It is Wright's test. The procedure is similar to that in the Tularemia diagnosis. Patient's serum is twofold diluted from 1 to 50 until 1 to 800s. And it is mixed with diagnosticum. It is colored again. Brucella are small, that is why they are colored. And after 1 day of incubation, you can see, you can see transparency above the layer of agglutinate on the bottom. You can see transparency. You can see turbidity with the purple color, uniformly purple. So it means that the test is positive until the titer 1 to 100s. Diagnostic titer in brucellosis for agglutination Wright's test, agglutination, lies between 1 to 100 to 1 to 200s. So we can say that our result is equal to diagnostic titer. The test is positive. The titer is 1 to 100. That helps to confirm the diagnosis of brucellosis. Questions? Do we follow?
Yes.
Okay.
Yes.
Yes,
That's
I
it.
don't have a
Good.
question.
Let us move on. Treatment includes antibiotics, effective against intracellular parasites. They include tetracyclines, aminoglycosides, and fluoroquinolones or fluoroquinolones. It's synonymic. And in chronic cases of brucellosis, because acute periods, primary steps of disease could be missed due to misdiagnosis. I told you that it resembles influenza in the beginning. Well, killed vaccine can be employed for treatment of chronic cases. The principle is already mentioned, discussed. Immunoprophylaxis for people with the occupational risk of disease, having animals involved in the processing of animal products, must be vaccinated against brucellosis. Live vaccine contains live bacteria of species Brucella abortus, BA. But it prevents infection caused by any species of Brucella because it induces cross immunity against Brucella because it is made of bacteria containing O antigen with A and M subunits that we discussed before. And it is effective for prevention of many cases of brucellosis caused by different species of Brucella because it induces cross immunity. Well, and it induces prolonged duration of immunity, by the way, that lasts up to 5 years. Well, measures of prevention measures include vaccination of animals that stop the transmission of parasites from animal to human, proper cooking, pasteurization of milk, control animal products, and good manufacturing practice, GMP standards. The last but not least, anthrax causative agent is caused by Bacillus anthracis that belongs to genus Bacillus, family Bacilliaceae. Here is a picture that shows different morphologies, different methods of detection of bacilli, by the way. This is a gram staining result. They are purple color bacteria that form chains with square, which are square-ended, very long, big rods in long chains with square ends. Here's a fuchsin staining that shows the presence of capsule. We can see encapsulated bacteria. You can see red color cells, again, pink background produced by specimen. And you can see transparent area, clear zone around the cells, that is capsule. And here is the result of Angeski staining used for detection of endospores. It is endospore staining. But unfortunately, this picture is not quite correct because Angeski staining helps to allows coloring vegetative cells. But you can see that it remains colorless, unlike endospores become colored. Morphology and tinctorial properties. They're gram-positive big rods with square ends. They form long filamentous chains that resemble bamboo sticks. Form endospores with a central location inside. In gram staining, of course, it remains colorless. It is oval in shape. Bacteria are encapsulated. They possess polypeptide capsule, and they are non-motile, unlike soil bacteria, soil bacilli. They are non-motile. This is one of the tests for differentiation. Used for differentiation. They aerobic, easily grow in a culture media. In a liquid medium, they grow on the bottom because they are too heavy, and they go down, forming sediment, leaving the supernatant fluid above, transparent. While growing in the culture media, in the solid culture media, they form big colonies with regular edges, with a rough surface that resemble medusa head or lion mane. Unlike soil bacilli, like Bacillus cereus, they are non-hemolytic. That's weird. That so such pathogenic bacteria, such dangerous bacteria, are non-hemolytic. In the right, on the picture, it is Bacillus anthracis. In the left, Bacillus cereus, and you can see similar appearance of colonies, but Bacillus cereus is hemolytic bacterium. Here are some tests used for differentiation of bacteria. First of all, in the presence of bicarbonate, they form smooth colonies, S colonies, because they become encapsulated. Then, in the presence of bacteriophage, they are lysed by specific anthrax bacteriophage. Here's appearance of colonies. And there is a very interesting test, which is known as a string of pearl test or pearl necklace test. What is the principle? They are gram-positive bacteria which are very sensitive to beta-lactam drugs, and they're destroyed by penicillin. Penicillin causes formation of spheroplasts. Spheroplast, I remind you that it is a sort of L forms of bacteria. When they form spheroplasts, they become oval or round, that under the microscope looks like string of pearl. So they can be cultured in a culture medium containing penicillin. And unlike soil bacilli, they are sensitive to beta-lactam drugs. They are sensitive to penicillin. And they change their morphology. They become L forms, spheroplasts that is easily detected under the microscope. I want to stress that they do not form strings on the agar. It's a specimen under the microscope. Biochemical features. They're highly biochemically active. There are some tests that help to differ between Bacillus anthracis and Bacillus cereus and other soil bacilli. You can see that Bacillus cereus are hemolytic. I already mentioned that. Bacillus anthracis is lysed by specific bacteriophage. Bacillus soil bacilli are motile, unlike Bacillus anthracis. And Bacillus anthracis is positive in strain of pearl test. They possess catalase activity. They possess sugar-lytic and proteolytic activity. And there is one more interesting test known as a gelatin test, liquefaction of gelatin. You can see Bacillus anthracis inoculated into the stub of gelatin, causes liquefaction, causes lysis of gelatin, destroys it. That resembles inverted fir tree. It is inverted fir tree phenomenon. It remains.
Guys, please switch off your microphone. Guys, switch off your microphone.
Thank you. So you can see this tube is inoculated by stabbing into the bud, and bacterium is non-motile, so it remains near the stick, near the inoculated area. And as it is gelatin positive, it causes liquefaction. It does it making it resemble fir tree, but inverted fir tree by layers. It is aerobic, that is why above the medium it makes the wider zone of liquefaction, and closely to the bottom the narrow zone. For the Bacillus cereus, for example, you can see complete liquefaction. It looks like that. It is semi-solid medium, but gelatin is a protein, and Bacillus cereus destroys the protein, and you can see it becomes liquid. So this phenomenon is helpful in differentiation of bacteria. Antigenic structure is by cell wall components, capsule antigen, which is protein, and it releases exotoxin that is protective antigen, one of the part of protective one of the part of 3. Exotoxin. Yes. One of the part of exotoxin is known as protective antigen, and it is protective actually, and it is antigen. Virulence factors include not many: polypeptide capsule that protects from phagocytosis, and exotoxin that consists of 3 parts: protective antigen mentioned above, lethal factor, and edema factor. Lethal factor is tested by inoculation of animals. You can see that exotoxin is fatal to mice, rabbits, and guinea pigs that may die within 24-84 hours. Well, edema factors causes swelling, edema in a portals of entry, in the portals of inflammatory area. But I want to stress that these factors can work only synergistically. Being injected to animals alone, they do not cause anything. But in a combination, they cause fatal damage. Well, and it is known that exotoxin is a typical AB toxin. What does it mean? It means that B subunit binds to the target, and it activates A. It promotes entering A, induces entering A subunit to the cell that causes toxic effect. Resistance, as it is typical to endospore-forming bacteria, their vegetative cells are very sensitive to boiling, autoclaving, antibiotics. It is sensitive to beta-lactam drugs, unlike endospores that are resistant. And it is mentioned here that autoclaving kills endospores only within 40 minutes. And endospores of bacteria remain viable for a long time in the soil, for decades in the soil. And that is why they can be shared with the soil to the patient. Epidemiology of anthrax.
خانم زهرا باشایی لطفا میکروفونتو خاموش کن.
Sorry, how many say to you, turn off your microphone? Please turn off your microphone.
Thank you.
I think
Source of infection.
they can't.
Livestock, domestic animals, cattle, swine, horses. And I want to mention reservoir because bacteria may survive for decades in the soil because they form endospores. So soil is mentioned as reservoir in this slide. Among the routes of transmission, the main route is contact route during processing animal products through abrasions, through skin lesions, and conjunctiva by accidental touching to the eye. Then it may be shared by a fecal-oral route with contaminated foods. Unfortunately, bacterial endospores survive heating. That is why even in properly cooked meat, they may survive and they can be shed by alimentation. Respiratory route and via transmission via blood are also possible.
Excuse me, Zara Pasha, please keep quiet.
Speaking about clinical picture, there is a cutaneous form that is characterized with the primary lesion known as a carbuncle. You can see in the portals of entry, you can see even the area exposed to the pathogen, to the contaminated sample. I mean, lizard, for example, right? Well, you can see necrosis, area of necrosis. It is ulcer with exudate that is necrotized in the center with elevated edges. Well, painful. And after, well, when it heals, this black substance is removed. Well, here's ocular form, which is very dangerous. You can see intestinal form, pulmonary and septic form has a high mortality rate when patient may die within hours after contracting the disease. Immunity is both cell-mediated and humoral, and it is prolonged immunity. But reinfection may occur in rare cases, in some cases. Type 4 hypersensitivity develops. That is why anthraxine can be used.
Teacher, sorry, can you kick out?
Which one? Clinical picture, you mean?
No.
No, sorry.
Yes, this girl, Zahra Parsai, because many times we said like to turn off your microphone.
Turn microphone.
She didn't let.
So, starting from what item? From what point? Previous slide or the current one?
Yes.
This one.
Yes, sorry.
So there are different forms, clinical forms of zoonosis. You know this from the beginning, right? I told you that bacteria have no tropism, tissue tropism to human organism. So they invade any organ and tissue. And as the main mode of transmission of anthrax is contact route, we can find in the portals of entry of parasite, we can find the typical local lesion known as ESCA or carbuncle. It is an ulcer with an exudate inside, that is with a necrotized tissue in the center, that is healed, leaving the scarring after that. Well, and here portals of entry, you can see maybe it is due to fur coat made of made of diseased animal hide. You can see it touches to the diseased hide of an animal here and here, and this is due to accidental touch to the eye. It is ocular form of anthrax. By the way, it was mentioned in the previous slide that infection is mainly observed in people with at risk of occupation, basically in people involved in the processing of animal products like leather. And you can see on the hands, yes, this infection, this inflammatory area. But primary lesion is this one. It is carbuncle. And it may be cutaneous form, like it is shown here. It may be ocular form, and pulmonary and septic form. The last three are fatal to a patient within few days after exposure.
Zahra, turn off your microphone.
Sarah, are you having fun by disturbing us? Are you having fun by disturbing us, Zara? Turn off your microphone. Otherwise, get out of the group.
Guys, calm down. I don't think so. She can hear you. I think she just like, okay, yeah, she can't hear you.
Immunity to disease. You can see that it is both due to cell-mediated and humoral immunity, which is prolonged. Because bacteria release exotoxin with a protective antigen. So such protective antigen induces antitoxic immunity development, humoral antitoxic immunity. But they also have capsular antigens that induce cell-mediated immunity. So immunity after recovery is prolonged with prolonged duration. But unfortunately, in some cases, occasionally, it may it may induce reinfection. Reinfection may occur. Well, and type 4 hypersensitivity may develop. Well, microbiological diagnosis. We are very close to the end. Shake up. Specimens include wound exudate, pus from local lesion from carbuncle, sputum in pulmonary form, feces in intestinal form, blood in systemic disease, I mean septic form. Urine sample can also be tested, and CSF. By the way, in generalized cases of disease, there will be hemorrhagic meningitis, due to circulation of bacteria and passing through blood-brain barrier. They can invade meninges, causing meningitis. Well, autopsy material in fatal cases, because disease is life-threatening, and animal materials are also tested: meat, milk, bones, wool, hides, and others. Methods, all 6 methods again are mentioned here. Rapid test, because infection is very dangerous. And here's a picture of immunofluorescence. You can see the specific morphology. You can see bamboo-like stick. You can see very long rods. We can see greenish glowing because it is immunofluorescence. Test direct immunofluorescence uses specimen test. It is a known antigen, treated with antibodies labeled with fluorochrome dyes. In our case, it is FITS fluorochrome.
Hey, who is this?
Hey!
Are you having Zara Pasha, are you mad?
Well,
Professor,
spectroscopical examination.
Professor, you can mute everyone.
Can you please kick her out, ma'am, if it's possible?
That's right. Okay.
Guys, you can't kick out in this application.
You can
I
mute.
know, but if there is possibility, then it's fine now.
No.
So while getting back to classes, yeah. So rapid tests include PCR, immunofluorescence, in vitro.
Oh, she's having fun with us.
Zahra!
Sorry.
زهرا پارسایی عزیزم میشه اینو ببندی